our experimental goal. How to design guide RNA, how to minimize the off-target effects, what is off-target effects, what are the delivery methods of CRISPR-Cas9, and the validation methods. How will we confirm that a knockout or a knock-in has been made? So without wasting further time, we will go into it one by one. So the first question is like how to choose the guide RNA binding region in a gene. A gene is very vast. It consists of exons as well as introns. So which region to choose for our either knockout or knock-in? So depending on knockout or knock-in, the site in a gene varies. Which site to choose? So first we have to decide our experimental goal. What is our goal of the experiment? Is it knock-in based experiment? Is it a knockout based experiment? Knockout is simple, like relatively simple, where you know that you are targeting a gene and you want to silence that gene. So you will just create a guide RNA. I will explain it later in the subsequent slides, but basically you will have to design a guide RNA and it will go and cut the gene at a particular site, which will cause a frame shift, causing the gene to get silenced. It will not code for the protein, or it will code for a different protein because of frame shift. In knock-in, there are different parameters. So you want to make a knock-in in which region? You want it to be done in a specific gene, or you want it in intergenic spaces. We all know that apart from genes, there are intergenic spaces where there are non-coding RNA regions. So these regions produce non-coding RNAs. So we all know about microRNA, long non-coding RNAs. You all guys must have studied. So if you want to do a knock-in of microRNA, then you target those regions. If you want to do a knock-in in a particular gene, so then you target the genes. In genes, which region? Exons or introns? So based on that, you have to define your experimental goal. In exons, there are again regulatory exons. Just a minute. There are regulatory exons and there are protein coding exons. So you guys must have studied that in a gene there are multiple exons. Okay, exons are the protein coding regions. So the first exon is always the regulatory exon. Exon 1 is always the regulatory exon, followed by the subsequent exons which are coding regions. Alright. So based on that, if you want to change the promoter region, then you will target the regulatory exon. If you want to change the protein coding region, then you will target the protein coding exons. So I've given few examples. If you want to tag a protein with a GFP, or you want to make a protein knock-in, let's say you want a protein to be fused with another protein, so that upon the mRNA produced, it will produce both the proteins from the same gene. So based on that, your experimental goal varies. And after, while doing the protein coding, while trying to do the knock-in and protein coding exons, depending on the N terminus or the C terminus, again it varies. Yesterday I was explaining to you what is N terminus knock-in, what is C terminus knock-in. So in that way, based on these factors, you have to decide your experimental goal. And based on this, the CRISPR-Cas9 based either knock-in or knockout, it varies. So let's first see how to choose the target region in a gene. Let's take an example of human GAPDH. Okay, imagine this is the whole genome, a whole gene of GAPDH, human GAPDH. You can see here there is a promoter followed by the first exon, then intron, then exon, then intron. So you can see here there are multiple exons here. Okay, so as you can see here, the first exon is always a regulatory exon. There is no ATG sequence starting from here. Whereas from the second exon, you can see that I have mentioned here, there is a ATG. So from here, the protein coding region starts. So this followed by this, and then here you will find the stop codon. Any of the three stop codons. Alright. So now imagine, as you can see here, once this, the mRNA, pre-mRNA is produced, once the RNA polymerase binds to the promoter, and after the pre-mRNA, there is RNA splicing. You all must have studied that in your bachelor's. So post-splicing, the final spliced mRNA is produced. This is the final mRNA, which is bound, to which the ribosome gets bound, at the 5' UTR after recognizing the cap, and then translation happens, which leads to the formation of polypeptide, which is nothing but the protein. Alright, so now imagine you want to do a knockout. So in order to do a knockout, which is essential, you want to basically silence this gene. Alright, so if you want to silence this gene, so which exon would you target? So if you target the last exon, it wouldn't much affect the the whole gene. Why? Because it is going near to the end. The protein's functional coding region might be in the middle or might be in the starting. So at the end, if you make a cut, there is no use. There won't be much effect on the human gap DH. That's why for knockout, the cut site is preferably near to the ATG site or whichever site which is highly functional, like the domains. There are multiple domains on proteins, right, which are highly catalytic, so which act as a catalytic site. So the cut site at those particular site is much favorable. Why? Because imagine if you are cutting just next to the ATG, the cut causes NHEJ, phased repair, which will cause formation of indels, insertions or deletions. Once insertions or deletions are formed, there will be a frame shift because the sequence has changed. The 3 sequence amino acids, we all know that amino acids, each amino acid is coded by 3 nucleotides, right? So that sequence changes. That's called frame shift. So because of that, the whole sequence will change. Once the whole sequence is changed, either the protein will not be formed or there will be some other protein, because it is not the same human gap DH which it used to be. So this is how knockout is done. Now if you have to create a guide RNA which is going to cut this site, so you will choose this site, okay, for knockout. I hope you guys are understanding this. Alright, if you have any doubt regarding this, you can ask me at the end. I will be explaining it to you very much clearly. So this is for knockout. Now next for knock-in. So for knockout, as you can see here, we are trying to do frame shift, but in knock-in, we are trying to do precise integration of a foreign gene into this already existing gene. Alright, so this is where the N terminus and C terminus comes into play. So as the ATG is starting from here, the protein coding sequence starts here. So once the protein is formed, this site will be the N terminus part, whereas the near the stop codon, this will be the C terminus part once the protein is formed. Imagine this is the N terminus and somewhere here is the C terminus. Alright, I hope you guys can picturize it in your mind. So if you want to tag a protein, this gap DH with GFP, green fluorescent protein. So if you want to do it, if you want to tag it, then you will either be able to tag it in the N terminus or in the C terminus. You might be thinking, why not just put it in the center? If we put it in the center, which is the main coding region, then the whole protein's ability will be lost. The GAPDH, which is a crucial enzyme in our body, its function will be lost because its coding region is cut into half, and then some foreign DNA is coming and getting integrated here. So because of which it won't function. So which will cause the cell to death. Alright. So because of that, it is not preferred to cut in between. It is either in the N terminus or in the C terminus. And that's why I have mentioned here. I have mentioned the site. It's N terminus knock-in or C terminus knock-in, site 1 or site 2. And one more thing for knock-in which everybody should keep in mind is it should always be in frame. Alright, when I'm saying it should always be in frame, which means that post knock-in, it should always be in frame with the coding region of the gap DH. Alright, I'll explain you that a little later, but I hope you guys can understand a little bit that what I mean by saying in frame, which means that the knock-in, let's say GFP codes for amino acids, lysine, threonine, arginine. So it should be in frame with the human gap DH sequence, which will be again proline, lysine, arginine. It should not shift. If there is a shift, then there is again frame shift, which is ultimately similar to that of NHEJ based. thing, so the function will be lost. So we don't want that. So in order to not go into that path, we should always make sure that it's always in frame. So once the knock-in has happened, either in the N terminus and C terminus, it will produce a fusion protein. I'll tell you about what is fusion protein in the later slides. So as you can see here, that these are the exons as I have mentioned here. So this is exon 1, which is a regulatory exon. This is exon 2, 3, 4, 5, 6, 7. Just an example. So here I am showing N-terminus knock-in. Why? Because I am choosing the second exon. Here ATG starts, and after ATG I am doing this knock-in of my gene of interest along with a selection marker. Selection marker will be an antibiotic resistant marker. Alright, this gene of interest is green in color. Let's say it's a GFP. Alright, so this is the donor macrohomology region donor construct. What are macrohomologies? As I said earlier in the last class, that macrohomology is these overhangs I had shown in my previous slide. I will show you once again. Here, this is the foreign gene, and this is the 5' macrohomology. This is the 3' macrohomology, which is complementary to the genomic DNA. Yesterday I had said this. So this region is complementary to this region. This region is complementary to this region. So when the cut is made, it will go on hybridize, followed by which the HDR based repair mechanism will act, and it will cause the integration of this foreign DNA into the genome. Same way, same thing I have shown here. Yeah. So this is the donor construct, donor fragment. We have our gene of interest here, followed which is to which the upstream part is the 5' macrohomology, which is complementary to the genomic DNA, and 3' macrohomology, which is complementary to the DNA, genomic DNA. So after cut is made, this will go and bind, all right, which will cause, and post repair it will cause the integration, which is knock-in, all right. And when the protein is produced, it depends. Either it will be a cleaved protein or it will be a linked protein. If you are linking this foreign gene, this foreign protein with your protein with a linker, there are protein sequences called linkers. There are flexible linkers, there are rigid linkers. You guys can go and read about this. It's a very interesting topic, as our topic is CRISPR-Cas9, so I am not explaining more about this thing. But it basically helps in linking two different proteins to each other. Flexible linker and rigid linker. Rigid linker provides distance, whereas flexible linker helps in the functionality of both the proteins. So depending on your protein type, you will choose either to put a flexible linker or a rigid linker. So this is for fusion proteins. Now if you want your GFP, green fluorescent protein, and the other GAPDH to be separate after the protein is coded, then you will add a T2A sequence. T2A sequence is a sequence which helps in cleaving off the whole protein into two. So after getting cleaved off, the GFP will become separate, the GAPDH will become separate. This is just an example image. This is not the exact image, but I hope you guys can understand from this that the T2A sequence, which is this red color sequence, it will get cleaved after translation, which will lead to separation of the GFP and the GAPDH. Alright, so moving forward, let's see about how to design a guide RNA. So